a differential expression today, and we're going into the different steps up until you get to that, starting from your raw FASTQ reads up till trimming, the alignment, and expression quantification, and then finally into differential expression and what types of parameters throughout this pipeline that you should pay attention to get the most out of your data. So, yeah, as I mentioned, so base pair, these are the topics that I'll talk about. Base pair offers multiple tools for alignment and as well as differential expression. So I'll talk a little bit about what are the best tools to use, what are the recommended tools to use. And then I'll talk a little bit about the different parameters in these steps and alignment and expression quantification and parameters you might want to pay attention to depending on your data, the data's quality, and so forth. And then finally, going to differential expression and how to set up a simple just two-group comparison. And if you have some more complicated experimental designs you want to do, I'll talk a little bit about how you can set that up on base pair. So this is the general steps in the base pair pipeline. We start with trimming the data using this tool called FastP. It removes the poor quality parts of the reads and as well as removing any remaining adapter sequences that might be in it. And this step helps improve the data quality overall and makes more of your data more usable for downstream analysis. And then after that, we offer two different pipelines for alignment, tools for alignment, STAR and Tophat, Tophat 2 actually. And I'll talk a little bit about the pros and cons of each of these, and the one that we would recommend that you use is actually STAR, but I'll talk a little bit about that later. And then after alignment, we have a FeatureCounts tool to quantify gene expression. And then after that, of course, you've got differential expression where you can offer two different tools again, a DE-Seq2 and Cuffdiff 2. And I'll talk about the pros and cons of each of those and what you can do with each. And then briefly, I'll talk a little bit about the gene set enrichment analysis tool that we use. So GSEA is a tool that's was published by the Broad Institute, and it's probably one of the most frequently used pathway enrichment analysis tools for RNA-Seq. And I'll show on base pair the results of that and what that looks like and how you can make sense of it. So then, yeah, I guess I can, let's see, let me share my screen here.
I'll briefly jump in here because I forgot to mention this while Charlie is sharing the screen. If you guys have any questions, just type them into the Q&A at the bottom, and then we'll answer that once the presentation is over.
Yeah, definitely. So, is that visible, Janetta?
Yep.
Okay. So this is, when you sign up for base pair, this is the dashboard that you'll see, and it'll show an overview of your different pipeline, your projects, samples, and so forth. And I'm in this project called example data. So whenever you sign up, we have existing example data loaded into your account. And if you just click on here, samples, and you can see those different samples. And here's a bunch of RNA-Seq data here, the bottom 6 samples here. And we also have some ChIP-seq and DNA-seq and single-cell RNA-seq samples as well for you to explore. So I'm not going to spend too much time talking about the basics of base pair and how you upload samples and so forth. I'll just quickly go through it, but of course, if you have questions, you can contact us. So you have your samples here, and once they're uploaded, after you've clicked this upload button, you just kind of click and drag your files here, and you can click it per sample. You can actually upload dozens of samples at a time, and it should automatically figure out which files correspond to which samples. So you can do that with here. So once that's done, your samples will appear here, and then you can start running analyses with these. And we offer a variety of pipelines here. You can click, and I won't go through all this, but the one I'm talking about, of course, today is the RNA-Seq steps, and you can go into detail of these steps and see what is actually done in these pipelines. So that's there for you to explore. So then to actually set up an analysis, you just click here, go to new analysis, and the first thing you want to do with your data is actually just run the expression count star, or you can also run the Tophat pipeline. So both are available. You can click, and then you just, again, click which sample you want. This one, for example. And that's the basics of how you get your sample uploaded, and you can set up the analysis. So, and as I mentioned earlier, back to this presentation, we offer two different alignment pipelines, one being Tophat 2 and the other being star. And this is a simple table showing some basic comparison among them, when they were published and how often they're cited, and some key features of them. In addition to alignment, they also can discover novel junctions for your genes, and both of them offer modules for gene fusion detection. So they can do a variety of things. And the key thing to note is actually that star has been shown to be more accurate compared to Tophat 2, as well as significantly faster. So we do recommend that you use star for your alignment on base pair. And we do keep Tophat, though, just for kind of legacy reasons. Some people still specifically want to use Tophat, so they're available if they want. But if you have no particular preference, I would definitely recommend using star. And then, so going into a little bit about the parameters, so going back to this page, when you have this set up, there will be this little button here called Change Default Options. And you can just click on it, and it'll show a bunch of parameters that you can change. And we try to provide some good default parameters for the analysis, but some in particular you may want to pay attention to. So the ones that I will talk about are multi-mapping, and also if you want to find out a little bit more, you can hover over this little question mark, and we've got some additional information that you can see. So multi-mapping and trimming quality threshold, as well as some other trimming steps that you can apply. So of those parameters, I came up with a little table here to show you what they are and why you might want to change it. Starting with multi-mapping. So what is multi-mapping? It's when you align your NGS reads to the genome, ideally you want the reads to map to one position. These are so-called uniquely aligned reads. And that tells you unambiguously that read came from this gene. But some case, in most, if not all samples of RNA-Seq, you get sometimes what are called multi-mapped reads. That is, you have a read that can map to multiple genes and with a kind of equal quality score. And it's unclear from which gene this read originally came from. And that can impact downstream analyses because it's, in terms of quantification, you don't want to count reads that didn't come from the gene. So by default, in the RNA-Seq pipeline, we discard those reads from multi-mapping. But I have seen scenarios where maybe you want to keep them, keep the multi-map reads. I've seen, like, for example, you've got some transgenic gene inserted into your sample, and maybe you just want to verify that the transgene is actually present within the sample, and maybe multi-mapping, it only show up when you include multi-mapped reads. You know, that's one example scenario I've seen with the project I've done on, and because if I discarded multi-mapped reads, that transgene didn't show up. Another reason you might want to include multi-map reads is if you're doing small RNA-Seq, and that is actually what we do for our small RNA-Seq pipeline. So just some background, those small RNA-Seq are just the microRNAs and other types of very small RNAs, classes of small RNAs that occur. And pipeline, we have a pipeline that's tailored to that type of analysis. And since, like, say, microRNAs, for example, are like 20 or so base pairs long, the number of multi-map reads can be very high. So for that reason, we keep the multi-map reads in the analysis. And there's even other things you can do, more sophisticated ways of dealing with multi-map reads that I won't go into, that have been used for small RNA-Seq as well. So multi-map reads, and then talking a little bit about trimming quality threshold, we have, this is for the trimming step, where we want to remove portions of the read that are poorer quality. And how it does this essentially is because in your NGS reads, each base pair in the read has a quality score associated with it. And you want to remove those parts that are poorer quality. And so it'll look at a window of, say, like 10 base pairs and compute the average quality of the base pairs in that region. And if they fall below this threshold, it'll cut out that portion of the read. And this trimming is pretty much something you do for any NGS data, and including RNA-Seq. But we do employ trimming for our RNA-Seq pipeline, but I guess you could say it's a light amount of trimming. And you might want to change this threshold if your data is of poorer quality, and you might need to do a more aggressive trimming for your analysis. And I'll show you here, this is that parameter here, trimming quality threshold. The default we use is 10, and maybe you might want to increase this to maybe 20. And I'll show a little bit later in how the trimming quality threshold can impact downstream RNA-Seq analysis. So this is just one thing to consider. And let's see. Then finally, I'll talk about a couple of these, this third row in this table here, the trim front one, trim tail one. That just refers to the top two are from the forward reads, and if you have paired end sequencing, the bottom is for the reverse reads. And all that does is it removes a certain amount of the beginning or end of the read. And if you set it to, like, if you set trim front 1 to 5, it'll remove the first 5 base pairs in your forward read. And this is something that's probably pretty infrequently used for RNA-Seq, especially for standard RNA-Seq. But if you know, for example, you have some non-standard adapter sequence that wouldn't normally be removed by the trimming step, you can specify one of these parameters, and you can say to remove the first 5 reads of the forward read, the first 5 base pairs of the forward read, because I know that that is an adapter sequence or some other kind of sequence, artificial sequence you introduced. So that's also there as well. And but also keep in mind, I mean, a lot of these parameters are good for standard RNA-Seq, and you shouldn't really need to change it for most of the time. But again, it does depend on your data set. So yeah, I showed you this before. So these are all the parameters you can. Then moving on to, I mentioned earlier, talking about how trimming actually impacts the downstream usable data. Here's a blog post we made that's available at this link down here, where I studied the impact of different trimming quality thresholds on how much usable data you get. So this is Q is what means is the trimming quality threshold, and it goes from 0 to 30. And if you recall, the default value is 10, and the y-axis here is how much usable data you have from after trimming. So this bluish color is what are uniquely mapped reads. So that's the ideal. That's what you want. You want all your reads to be uniquely mapped, and that's a significant portion of it. But you also have in green, which are the multi-mapped reads. You don't want those. That means it's hard to tell from which gene a read came from. And the worst case scenario you've got in red at the top are the unmapped reads. So those are reads you can't use at all. And what this figure shows is that as you increase the trimming quality threshold, you get more uniquely aligned reads. The difference is subtle, but it makes an impact for your downstream analyses. So it's something to take into account, and it's why we have a moderate amount of trimming threshold. But there is a downside to this, that with more aggressive trimming, you can actually start to negatively impact your analysis. And that's what, in this example, the negative impact is very minor. And again, the x-axis here is the Q threshold, so the trimming quality threshold. And the y-axis is the correlation with the gene expression data from the untrimmed data. So as the trimming quality threshold increases, the correlation decreases. So in this example sample data set that I used for this, the impact is basically nothing. But in certain situations and poorer quality data, you can see this correlation change quite a bit. So that's the only thing to keep in mind if you want to use a very aggressive trimming threshold like 30. And then, yeah, so those are those parameters for the alignment step. And you can just, once you're done, you click run analysis and it'll go through. And this pipeline should take 30 to 60 minutes to finish. So it's fairly quick. And once it's done, you'll get an email and you get a link and it'll show up here in your analyses. So, and down here are those expression count star, that's the alignment pipeline. And I'll just go into one example. And this is a basic report of the data, showing at the top here the quality score threshold. So you've got before and after trimming. And this is, the quality, the mean quality per position in the reads. So in these reads, they're around 100 base pairs long. And everything, you generally want quality scores above 30. Then after that, it'll show the different alignment steps and using this Sankey plot. So starting, there's around 19 million reads in this sample. And after trimming, it removed about 0.04% of the reads because they were poor quality reads. So this is actually a fairly good, it is a very good data set. And then it shows you the alignment statistics, and it shows 88% of the reads are uniquely aligned, and that's good. Whereas you have around 10% multiple aligned and 2% unaligned. So again, this is a fairly good data set. You generally want to see a percentage of uniquely aligned reads in the 80s, 90%. And this percentage of uniquely aligned reads should increase as you have longer reads as well. Whereas if you have shorter reads, like 50 base pairs or even shorter, that the percentage of multiple aligned reads can really increase. So, and then, yeah, at the end of the analysis, you have 18,500,000 or so reads usable. And again, some more statistics here later showing the percentage of reads that map to different features. You've got the mRNA, intronic, intergenic region. And finally, these two figures show the quantification of the sample. So we provide multiple per gene expression quantifications, or as the raw counts, the FBKM and the TPM. For all our users. So, and also, of course, a very nifty feature here at the bottom is the genome browser. This is basically IGV for those who have used it, except this is embedded in the browser, and you can, navigate around this and see expression of the genes that you're interested in. So then on to differential expression. So again, here's another simple table showing what the two tools that we offer, DE-Seq2 and Cuffdiff2. When they're published, how many citations, and as you can see clearly, DE-Seq2 is a very, very popular tool, and it is actually something we recommend that you use. But we have Cuffdiff2 of it on our site because some people have requested it, and so it's available there to use if you want. And DE-Seq2 is really just overall a better tool. One of the things it can handle is more complicated experimental designs. While Cuffdiff2 can handle time series data if you have it, it can handle, DE-Seq2 can handle other multi-factor experiments and so forth. And I'll get into that a little bit later, what I mean by that. But also some benchmarks have shown that Cuffdiff2 is often a little too conservative in finding differentially expressed genes, and just has a general lower ability to find true differentially expressed genes. So again, we recommend DE-Seq2 because it's more accurate and it's just more flexible in terms of the types of analysis you might want to do. And so I mentioned earlier, like, how do I, what are the types of experimental designs I can set up with your data? And starting here on the left, the simplest design is the two-group comparison. And I'll show you that on base pair and what that looks like and how you set that up. Then I'll go into the other types. So again, you can go to the analysis, new analysis, and click on DE-Seq2. And you have your two groups here, treatment and control. And you can just click here, click all the samples that you want. And they are there. And that's it. That's how you set up a just two-group comparison for differential expression. But what if your data is a little more complicated than that? What if your experiment design was a little more complicated? So we do offer other ways to set up the analysis. One of them being like a three or more group comparison. You can actually do up to 12 groups. That's in this second one here. Now, what this actually does, we have a pipeline for this, is it actually compares, for example, group 1 versus group 2 and 3 combined. So it finds what's unique in group 1 compared to all the other groups. And similarly, then it goes to group 2 and compares group 2 versus all the other groups, and so on. It'll do that for 3 to 12 different groups that you have. And again, I'll show you how you set that up. So again, you can just pull up GDC2 3-group comparison. And again, you have 3 different groups that you can set up here. You can click on your samples for each of them. And that's that. And you'll notice I put just one sample in this one group. That brings up an important note is when you do these comparisons, it's highly advised to have at least 3 samples for each group. It just leads to better statistically significant results. And it's just a better way to do science if you have multiple biological replicates in each group. But it can handle one sample in each group, but it's not ideal. So that's 3 groups, and you can just add more groups if you want, and so forth, up to 12 groups. So again, that'll find, it'll compare each group versus all the other groups. That's how this analysis works. And then how about these 2 types of analyses on the right? What if you have a paired analysis? So what is a paired analysis? That is, if you have 2 different groups, say you have a group of patients and you took samples from them at like time 0, and then you gave them a treatment or something, and at a later time point, you sampled the same patients. So it's important to, when you're comparing this like baseline and this treated group, you should take into account the fact that you're sampling the same patients, twice. And you can do that with what's called a paired analysis. You tell the analysis how you set it up. You tell it which samples correspond to the same patient, and this leads to a more powerful analysis. You have a greater ability to find differentially expressed genes when you give it that information, and it's otherwise, if you don't give it that information, you often will get fewer significant results. And so how do you set that up? It's fairly simple, actually. So again, you just go to the 2-group comparison, as I mentioned earlier, and add your samples. And then you can click on this change default options. And what you'll see here is what's called a factor. And this will always here by default. You can't change this because it compares to 2 different groups. So what you'll have to do actually is add an additional factor. And this is where you can enter your information and tell that this is patient 1, this is patient 2, patient 3, and this is patient 1, 2, and 3. And what this analysis will do is it'll now take that into consideration for when it's doing its differential expression results, and you'll get better results. So that's how you set that up. And I should also say, this can be anything. It can just be and it could be, just any kind of text. As long as it, matches, there's some correspondence between the 2 different groups that this occurs twice and so forth. So it can be whatever you want, really. So then finally, the multi-factor comparison. And what is that? That is, if you've got, say, 2 different groups of samples, say different mice, and you applied a treatment to one of them, and maybe actually your samples are a mix of male and female mice. And that can really negatively impact the analysis because that introduces a lot of variation within each group, transcriptionally within the RNA-Seq data, depending on what you're studying exactly. But this factor can be anything, male or female or whatever. But the point is that it's an additional source of variation that you want to remove from the comparison. You want to focus in on what are the differences between group one and group 2. You don't want to focus on what are the differences between the male mice and the female mice. And again, how do you set that up? Again, it's very simple. You just type it in and say, okay, these two are male and these two are, that one's female, and this one's female, and that's it. And again, you don't have to say male or female specifically. You can use whatever words or letters you want. But that's how you set that up. And when this analysis compares a group 1, group 2, it'll take the male or female into consideration. So, and then those are some of the more interesting comparisons that people often request at base pair, and that's how you can set that up. So then I'll go on, I'll show you, there are other parameters here as well, if you want to change around for those who maybe know a little bit more detail about differential expression. Like you can change it from WALD to log likelihood ratio test. You can change the P adjustment method, and so forth. There's other things you can change as well. So then showing you what this looks like after the analysis is done is, I'll show you here. So again, the analysis should be very quick, probably half an hour or less, and you'll get an email and you'll see this figure, these figures. So this is just showing you a volcano plot. This is plotting the log-fold change against the p-value, the negative log 10 p-value. This is a heat map of your samples, of the significantly differentially expressed samples. And you can change these p-values and so forth, and it'll update these figures. And you can even download the figures as PNG or SVG, and you can put these right in your papers once you're satisfied with it. And here's the actual list of differentially expressed genes. And you can sort it by the p-value or p-adjusted value and or log-fold change. And is a note to make is when you're deciding what are your differentially expressed genes, we recommend that you use the p-adjusted column. This is a better way, a better metric compared to the p-value. The p-adjusted just reduces the chance that a given gene is a false positive. And so you should definitely focus on this. You can, again, you can use thresholds like 0.05 or 0.1 or 0.01. Those are all fairly common. There's no real absolute best threshold to use, just those are very common ones. And so that's the list of differentially expressed genes and what it will look like. So then I'll talk a little about like, how do you make sure your data is of good quality? So this is an example figure. I showed you these figures on the right. And we also include a PCA plot. And that's actually right here in this figure. So a PCA plot is just like a dimensionality reduction technique. It shows you on a global scale, how do your samples cluster together? And the ideal scenario is that your two different groups of samples will cluster, separately. And in this case, you've got the doxycycline and the untreated samples. So you can see the doxycycline samples are over here, and your untreated are over here. And that's usually what's expected. But maybe in this scenario I'm showing on the slide, maybe you found that there's some mixing of the samples. And in this sample, say there's a red dot over here. I just drew these circles to kind of highlight that they're two different groups. And say you found that there's a red dot over here over with the blue dots. And that's unexpected because maybe it's part of that group and you would expect it to group with those other samples. So this could actually be from multiple causes from this. It could be that maybe you just mislabeled a sample. And if you did, you can easily run another analysis and fix it, and that's not an issue. Or it could be that maybe your sample is actually an outlier, that it just doesn't conform with the other samples in the group for some biological reason. And in that case, you actually might want to consider removing that sample. It's just doesn't necessarily help you find the differentially expressed genes between your two groups. So that's a fairly common thing to do. You remove outlier samples. And so and one thing to note is that the figure on the left is the results from this potentially mislabeled sample. And as you can see, this heat map looks a little strange. You would expect that the samples would group pretty nicely together by group. But you've got this sample here on the right that seems to not match very well with the other three samples here. So assuming this is just a mislabeled sample and I ran another analysis, and here's how it can change from the mislabeled sample figures on the left to the correct labeling on the right. So the biggest thing to take notice of is on the right, you've got around 300 differentially expressed genes, but on the right, with the correct labeling, you've got around 1,500 differentially expressed genes. So this shows you, if you have a mislabeled sample, it can really negatively impact your analysis and make it much harder to find your significant results. And you can actually have the same scenario if the sample maybe wasn't mislabeled, maybe it was an outlier, and so that's why you should consider maybe just removing it. So that's PCA. We provide a PCA analysis so you can do those types of diagnostics of your data and check if it makes sense. Because you would expect the samples to group together by group. So then moving on to the GSEA analysis is the final thing that we offer for this pipeline, shown here at the bottom. So by default, we use the GO pathways to do enrichment. And so these two different tables show which pathways are enriched in each group. And again, here at the top, it's some organophosphate ester transport system, and that's what's most significantly associated with this group. And maybe for the untreated, you've got positive regulation of fibroblast proliferation. And this is sorted by the most significant results, and I'll go through quickly what these different columns mean. And this is, of course, the pathway name. This is the number of genes. The size is the number of genes in the pathway. And ES and NES, that stands for enrichment score and normalized enrichment score. You can mostly just ignore those columns. The software uses those internally to compute the p-values and FDRs. And then onto that, so you've got the nominal p-value. That's what this stands for. And again, you've got the FDR Q value. So this is the most important column to pay attention to in terms of determining which pathways are significant. It takes as input the nominal p-values and does a correction so that any given pathway is much less likely to be a false positive. And so this is the column to pay attention to. And again, you can use thresholds like 0.01 or 0.05 or 0.1. I've seen all those to determine what are the significant pathways. And the FWER, family-wise error rate, is a p-value is similar to the FDR p-value, but it's much more conservative and it's generally not used. So you can ignore that one generally. And then the rank at max, it's a little more complicated to get into, but it's usually not a hugely important statistic to pay attention to. So then, yeah, and these are all the results, and you can click it and go through it all, and you can even download it as a CSV if you need to, and it's all there for you. I think that's about it. Yeah, so then that's it, yeah. And thank you for listening. I will do a Q&A session now. So there's a little chat icon. You can ask questions and I'll spend a few minutes here answering them. And so someone asked, what does STAR stand for? So STAR is the tool we use for alignment. I'm actually not sure. That's something that can be pretty easily looked up though, I'm sure, on the publication. So you can just Google that. If you are mapping to dikaryotic genome or the genome has many homologous genes, would you suggest to discard multi-mapped reads? In that case, you might consider actually changing some of the parameters to reduce the amount of mutations that are allowed within each mapping. That's one solution. And it is worth running the analysis while using the multi-mapped reads and checking how those things work, how those results look. But in that case, when you have many homologous genes, that is the difficult situation with multi-mapped reads. It's harder to determine from which gene it came from. But so reducing the amount of mismatches or so that are allowed in the mapping is one thing to consider for that analysis. And it is worth looking into including the multi-mapped reads. Why are there unmapped reads? So this could be from a variety of reasons. It could be that maybe it was ambient DNA when the sample was prepared, so the RNA could actually not be from the organism, and so it couldn't map to it. It could be that the RNA, the read was too poor quality. Otherwise, it could have too many what are called ends in the read. A variety of reasons why you might have unmapped reads. And it could also be that it's too short and the aligner won't align it. So there are a variety of reasons why. What about DEC comparing to EdgeR? EdgeR is also a very popular tool. It's actually quite similar to DEC. It's the underlying statistical model. I haven't read up on EdgeR recently, so I can't be 100% sure, but It has a slightly different statistical model, and I believe I've seen comparisons between the two tools, and DEC2 outperforms at least a little bit, but I'm not 100% sure on that. I haven't used EdgeDAR in quite a while, and so. Can DEC differentiate between different splice variants? No, it does not do that. Cuffdiff can, though. It will do a differential isoform expression. There are other tools to do that sort of thing. But one thing we do offer actually is you can set if the differential expression should be done at the gene level or the transcript level, the transcript isoform level. There is a parameter for that you can change. Though it's not really recommended, it's better to do, because DEC2 is designed to do differential expression at the gene level, not the isoform level. Because that just opens up a whole set of issues in terms of like reliably assigning reads to which isoform. But it's the options there if you want to try it. What is the cutoff for a significant FDR Q value for GSEA? You can use 0.01, 0.05, 0.1. I've even seen 0.25. There's really no best answer. It's just one of those things that use what publications use, and the ones that I gave are ones that I frequently see in various papers. There's no real way to define what's the best way, but something like 0.05 is a good start. In the case of experiments when looking at infected cells, is it possible to include a custom chromosome for RNA from the pathogen? And how would you suggest going about that? It is possible. One option we can do is actually include that custom chromosome as a spike-in for the analysis. And you can contact us using the chat icon or email us, and we do charge a small fee for adding genomes like that. But we have the pipeline set up so that it'll align the reads to that pathogen, that spike-in genome. It'll quantify expression of the genes, if there are any, and as well as you can include it in the differential expression results. Our pipeline will output what genes are differentially expressed in that spike-in genome. And in that case, that spike-in is the pathogen. So that is certainly an option. You can reach out to us, and we can add that genome to our genome offerings. Are these softwares on base pair free to access? We do offer a free trial. There's free to sign up, no strings attached. You get 15 days of free access, and you can upload 6 samples. But there is a limit in what you can download from the software unless you set up a billing account. So, but otherwise it's free to try. You can, as I mentioned earlier, there's example data loaded in the account, so you can check it out. So definitely do that. And what else? So someone asked, does the comparison pipeline support greater than 3 treatment groups? Yes, it does. There's a DEC 3+ comparison, and you can have up to 12 groups to compare. And again, what that'll do is it'll compare each group against all the other groups. So it'll find what genes are unique to that group compared to all the other groups. And someone else asks, can it use a de novo transcriptome as a reference? So we do actually offer Trinity as a de novo transcriptome assembly. So you can certainly use that. But as far as adding it as a reference genome to our site, that's a different matter. Again, we would charge a small fee to add it to our site. And you can reach out to us for that. But otherwise it's not feasible fully on the user end. We would have to get involved a little bit to help you do that. So certainly reach out to us. Okay, and I think that's all the questions. Thank you again for attending. And we do these webinars once a month, so certainly keep an eye out for emails from us if you want to attend another one. And if you have more questions, please feel free to reach out. You can, we have a little chat icon at the bottom, and you can just open it and ask us questions, and we'll get back to you very quickly, or just email us. So it's great. Thank you all.
Thank you, Charlie. Yeah, and thanks everyone for attending. We will send the recording to those of you who might have joined halfway through the webinar. Stay tuned for that in a couple days.
Okay.
All righty.
Bye.
Bye-bye.